So, Liam, ever feel like you're solving a jigsaw puzzle and suddenly realize a bunch of pieces are missing?
Absolutely. It's not a good feeling.
That's what working with clinical trial data can feel like when there is missing data. And depending on how many pieces are missing, you might still be able to see the big picture. Like, okay, I think that's a picture of a sailboat. But if too many pieces are missing, or if the missing ones happen to be right in the middle, say, the sail, then suddenly you can't really make out the big picture. Or you might think the big picture is something completely different.
Welcome, everyone, to a module created by the HDRN Pragmatic Trials Training Program. I'm Liam, and I'm here with Angela. Today's module is all about missing data in pragmatic clinical trials embedded in routine care, where missing data is one of those unavoidable gremlins that every researcher will encounter at some point. We'll talk about why missing data might happen more in pragmatic trials, why it's such a big deal, and what we can do about it. By the end, you'll understand the different types of missingness and hear about strategies to prevent missing data in the first place, and how to handle missing data in your analysis. So, grab a coffee, but be careful, don't spill it on your data forms.
Before we get into it, let's first go over two of many examples where missing data seriously compromised the conclusions of large trials. Take the STAR-D depression trial that was funded at a cost of 35 million US dollars and enrolled 4,041 patients who screened positive for major depression. A major criticism was that patients were excluded from the primary analysis if they received citalopram, but dropped out before providing their final depression scores. Those missing cases were treated as if they'd achieved similar outcomes to those who stayed. That assumption likely inflated the reported remission rates and painted an overly optimistic picture of the treatment's effectiveness. A similar problem arose in the record trial evaluating rosiglitazone, which included about 4,500 patients and likely cost over 30 million US dollars, although the total budget was not publicly reported. Investigators noted an unusually low rate of heart attacks among control patients, a result likely driven by incomplete outcome ascertainment. With around 10% lost to follow-up, missing events distorted the true incidence rates. These examples show how missing data can erode validity, bias estimates, and leave even large, well-funded trials open to serious doubt.
Now, let's start with the basics. Missing data happens anytime we expect to collect a value, but don't. It could be as simple as a patient missing their clinic visit, a provider forgetting to complete a form, an event being misclassified as something else, or a lab result that never makes it into the system. Sometimes there's an equipment failure, or a participant decides to withdraw from the study. There are lots of possible reasons for this, all leading to the same problem: missing information.
Exactly. And pragmatic trials are especially vulnerable because we aim to keep things as close to usual care as possible. That often means fewer required study visits and greater reliance on existing sources like electronic health records or administrative data, rather than detailed research forms. It keeps trials realistic and lowers burden, but that flexibility can raise the risk of missing or misclassified data, and the risk varies by data source. For example, in administrative data, all-cause mortality is usually captured well, whereas non-fatal outcomes like myocardial infarction can be missed or differentially detected depending on care contact and surveillance intensity. With fewer touchpoints and less structured collection, the chances of missing this naturally increase. Missing data is, in a sense, unavoidable, but it can't be ignored, which brings us to why it matters so much.
Missing data is a huge threat to a trial's internal validity. It can compromise how confidently we interpret results. It comes down to three major threats: reduced statistical power, selection bias, and information bias. So let's go through them one at a time.
First, missing data reduces power. That means your effective sample size shrinks, which makes it harder to detect treatment effects, even if they exist. Imagine planning a trial with 100 patients per group, but 20 in each arm don't provide outcomes. Now you're effectively analyzing only 80 per group. With fewer data points, your estimates become noisier, and the chance of a false negative increases. Think of missing data like a dimmer switch on your trial's power. More missing data means a dimmer statistical signal.
Second, missing data can introduce selection bias. This happens when the participants whose outcome data are analyzed differ in meaningful ways from those who aren't. For example, if those with missing information tend to be sicker or have worse outcomes. This is a problem even if the missing information is similar across groups. That's because the group you're analyzing is no longer the same population you randomized. In other words, your trial results might not reflect the effect on the original target population.
Even worse is when you have unequal missingness between groups. Imagine in a two-arm trial that Group A had 5% missing outcome data and Group B had 20%. That gap can signal trouble. Is Group B's missing data related to the treatment being harder to tolerate, or is it related to an ascertainment bias? When one group loses more data or has more opportunity for detection, differences in outcomes may reflect unequal missingness, not true treatment effects, weakening internal validity.
And third, missing data can create information bias, where incomplete or inaccurate data distort the very estimates we rely on. This includes ascertainment bias. For example, in routinely collected data, all-cause mortality is usually near completely captured, whereas non-fatal events might depend on the number of care encounters, coding practices, and clinical setting. Second is uneven data quality. Differential surveillance or care-seeking behaviors between groups can then produce group-specific detection bias that might inflate or deflate event rates for reasons unrelated to the treatment. In those cases, results start to reflect data quality rather than the true treatment effect. Finally, even small gaps can cascade through a trial, reducing power, altering who's represented, and ultimately eroding the accuracy and credibility of the conclusions. That's why preventing and properly handling missing data is so critical to maintaining internal validity.
Here's some good news. The best way to handle missing data is to keep it from happening as much as you can. Of course, we can't prevent all of it, but we can certainly try to minimize it by smart trial design and conduct.
Absolutely. Preventing missing data starts at the trial design stage. First, engage a qualified statistician early. Statisticians can help identify where data might get lost and design ways to prevent it, whether through simpler data flows, realistic visit schedules, or better tracking systems. Second, use smart scheduling. Keep study visits close to normal care and send reminders by text, call, or email so assessments actually happen. Third, streamline forms. Collect only essential data. Creating long, tedious questionnaires almost guarantees skipped items. Fourth, incentivize completion. Even small gift cards or a sincere thank you can improve response rates for patient-reported outcomes. Fifth, train staff. Emphasize why complete data matters and have coordinators double-check key fields before patients leave. Finally, embrace flexible technology. Allow data to be collected through phone or telehealth visits when in-person appointments aren't possible. Together, these simple strategies can dramatically reduce missing data and strengthen trial integrity.
Those are great tips. The bottom line is you want to plug the holes in the bucket while you can, instead of trying to refill water after it's leaked out. So investing in training, pilot testing your data collection procedures, and making the process as user-friendly as possible for participants can pay huge dividends. That said, your trial will most likely need to deal with missing data elements. Another design stage consideration is to specify in the protocol and statistical analysis plan how you plan to handle this missing data. This might sound like putting the cart before the horse, but it forces the team to confront the issue early. Regulatory guidelines like the ICH E9 recommend that trials predefine their approach to missing data and do sensitivity analyses for it.
Not all missing data scenarios are equal. The top box defines the missingness mechanism as the probability that an item is observed based on what we've recorded, what's missing, and our model settings. In practice, we assume one of three cases to guide analysis. First, missing completely at random, where the chance of being missing is unrelated to any data and it is likely equal chance across participants. Second, missing at random, where the chance of being missing depends on observed information, allowing modern methods like multiple imputation to fill in plausible values. And third, missing not at random, where the chance of being missing still depends on unobserved values, meaning the effect can't be learned from the data alone, and we need pre-specified sensitivity analyses to test how conclusions change under reasonable what-if assumptions. Keep in mind that differential ascertainment can show up as missingness, where an event is never recorded, or as misclassification, where an event occurs but is miscoded. Both threaten internal validity. Let's go into these in a bit more detail.
Again, missing completely at random means exactly what it sounds like. The data are missing purely due to chance, with no relationship to patients' characteristics or their outcomes. For instance, say a lab sample is accidentally dropped and destroyed. That blood test result is missing completely at random because the chance of that happening wasn't because of the patient's identity or their health status. From an analysis perspective, this is the best case scenario for missing data, aside from having no missing data at all. That's because the subset of patients who have complete data is essentially a random sample of all patients. That means our analysis is unbiased, even if we ignore the missing bits. That said, missing completely at random is kind of rare in practice.
Next is missing at random. Now, the phrase at random is a bit of a misnomer here. It doesn't mean completely random. It means the chance that a value is missing can be explained by information we've already recorded, not by the unseen value itself. For example, if younger participants are less likely to return a follow-up survey than older participants, the missingness relates to age rather than to the unreported survey answers. The phrase missing at random is an assumption about the missingness process, not something we can prove from the data alone. It's most credible when we've measured the key factors that drive missingness and planned our analysis to reflect them. Importantly, many commonly used imputation approaches assume the data is missing at random.
Next is the troublemaker. Missing not at random. This is when the reason data are missing is related to the value of the data itself. For example, suppose sicker patients experiencing severe adverse effects are more likely to drop out or be lost to follow-up. Their missingness might be related directly to our outcome of interest. With this type of missingness, even if we look at all the other observed variables, we can't explain away the missingness because the missing element might be tied to the outcome. Missing not at random is often called non-ignorable missingness, because you can't just ignore it or handle it with standard methods without potentially biasing results. So, with this type of missingness, we often use sensitivity analyses to test how our results might change under reasonable alternative assumptions about the missing data.
All right, so despite our best efforts, we finish our pragmatic trial and, lo and behold, some data are missing. Now comes the question, how do we deal with those holes in the data set when analyzing results? There are several strategies, like the ones listed here, each with its own pros and cons. So let's go through them.
The most basic approach is called complete case analysis, which basically ignores the missing data by analyzing participants with all the required data. For example, if the primary outcome is missing for 10 out of 100 participants, you'd analyze the 90 who have it and pretend the other 10 never existed. The trouble is, if a lot of participants have missing data, you could lose a huge chunk of your sample. As we discussed earlier, if the data are truly missing completely at random, then complete case analysis produces unbiased treatment effects, with the caveat that you'll have reduced power for detecting a treatment effect due to sample size erosion. Complete case analysis is generally not the ideal method if more than a trivial amount of data is missing. For example, if the observed missingness exceeds the attrition rate assumed in your power calculation, your effective sample size will fall below the target, and the trial may be underpowered.
Another approach you'll see with categorical variables is to add a missing or unknown category. It's occasionally useful for covariates or secondary outcomes. For example, including an unknown level for baseline employment, because it keeps participants in the analysis instead of dropping them. But treat it with caution. You're not recovering information, you're just labeling the gap. The unknown level can be hard to interpret, and if missing this relates to prognosis or treatment, you may simply encode bias rather than remove it. Statistically, missing isn't a real value and may not behave like the other categories. As a rule, avoid this for primary outcomes. Think of it as a pragmatic workaround for certain adjustments like propensity score models, not a robust solution to missing data.
Another common quick fix is single imputation. Fill each missing value with one guess. Examples include mean imputation, where you replace missing values with the overall average. Last observation carried forward for longitudinal data, where you carry the most recent value forward. Worst case substitution, which assigns the least favorable outcome to missing values. And regression imputation, where you predict missing values from other variables. The upside is that it is easy and yields a complete data set. The downside is that it builds in strong assumptions. Mean imputation reduces variability and produces confidence intervals that are too narrow. Last observation carried forward can distort trajectories. When missing this relates to outcomes or covariates, estimates become biased. The bottom line? Single imputation is acceptable for quick exploration, but it often produces biased and overconfident results and is not suitable for primary analyses.
Now, a more robust approach is using multiple imputation. Instead of filling each gap once and treating it as truth, we create several complete data sets by replacing missing values with different plausible values drawn from models informed by the observed data. We analyze each data set and then combine the results, so the variation across them reflects our uncertainty about what was missing. Under the missing at random assumption, this reduces bias and yields more reliable inferences than single imputation. It is not automatic. You need a well-specified imputation model that includes variables related to missingness and to the outcome, and you must ensure the imputed values make sense for each variable type. While implementation can be complex, modern software helps. When feasible, multiple imputation is preferred because it uses all available information and properly accounts for uncertainty.
Now to model-based methods. These approaches use all available data under explicit assumptions about why values are missing. First, maximum likelihood mixed effects models are common for repeated measures. You specify random effects and a covariance structure, and the model borrows strength across time so participants with partial records still contribute. This is valid when missingness can be explained by observed information, the usual missing at random assumption. Second, weighting methods reweight the complete cases to represent everyone. With inverse probability weighting, you model the chance a record is observed and upweight those who look like the ones you lost. With augmented inverse probability weighting, you add an outcome model, giving you double robustness if either model is correct. The strengths are efficient use of partial data and a coherent likelihood framework, with weighting adding robustness. The limits are that results hinge on correct model specification and on assumptions like the data are missing at random. However, if dropout is informative or truly not at random, estimates can still be biased, so diagnostics and sensitivity checks are essential.
Importantly, no method is perfect. You can't just hit a button and solve missing data without thinking deeply. The best practice is usually to do your primary analysis with a reasonable method, say multiple imputation or a mixed model under the missing at random assumption, and do sensitivity analyses using alternative methods, like a complete case analysis or an extreme imputation scenario, to see if they lead to a different conclusion. When missingness likely depends on the unreported values themselves, we can't learn the answer from the observed data alone. So, we stress test the same target of interest under reasonable, clinically motivated assumptions. Two practical tools. First, a pattern mixture nudge test. We complete the dataset under Missing at Random. Then, shift the imputed outcomes a little better or a little worse across a plausible range and pool the results. If conclusions hold, they're more convincing. Second, the tipping point and bounds. We systematically vary the assumed outcomes for the missing data, by arm or overall, and identify how extreme they would need to be to reverse the decision. We also show best and worst case bounds for context. The strength of these approaches is transparency. They make missing not at random assumptions explicit and quantify the robustness of the trial's results. The limitation is that assumptions are untestable, and results depend on the scenarios we choose, so choices must be clinically justified and clearly reported.
We've treated missing data like the villain, and it can be. But with smart planning and transparent analysis, it doesn't have to derail your trial. In pragmatic studies, the 1-2 punch is clear. Design and operations that minimize missingness, followed by careful, well-documented methods to handle what remains and openly acknowledge uncertainty. That's why we bring statisticians in early, train teams to capture complete data, and pre-plan how we'll analyze and stress test gaps. Think of it as insurance for your study's validity. That's a wrap for today's episode.
Thanks for listening. If you found this content useful, subscribe and click the bell so you'll get notified when new modules drop.
Goodbye.
Goodbye.